Hello everyone, welcome to the second session of Genetics Introduction. So we were discussing the different terminologies that is related to genetics. In the first session, we discussed the different structural organization of the DNA, namely the DNA double helix, followed by its more condensed state, which is called as, or more coiled structure, which is called as a solenoid, that can be seen in the left-hand diagram in this particular slide. So the topmost one is the double helical structure, followed by the beaded string structure, where the double helical DNA is getting associated with the proteins, as you can see in that particular video in the right hand side. It associates with the protein, forms the nucleosome. The nucleosome again coils in the form of a hexagon, as you can see in the diagram, which is also seen in the right hand side. And such diagonal structures are repeated one behind the other of the histone, the hexagonal histone structure, or the hexagonal histone portion, and that forms a rod-like structure. And that rod-like structure is called as a solenoid. And this particular solenoid is again coiling. So a solenoid will be a rod-like structure, and as you can see in the diagram, it will be a straight rod-like structure, which can be called as a fiber, etc. And such a fiber, or the solenoid, it again, or this particular structure, this red line which I drew here, consider it as a solenoid. That solenoid will again coil and form a much more shorter structure. That means it is getting more and more compacted, and this compaction will ultimately end in a structure which is called as a chromosome. And this particular thing happens only during the division phases, like either mitosis or meiosis. So whenever the cell is not engaged in division, that is whether mitosis or meiosis, whatever the case may be, whenever it is not involved in mitosis or meiosis, the DNA inside the nucleic acids, or the genetic material inside the cell, is usually present in the form which is called as the solenoidal structure. When a cell is at rest, that means the cell is at rest means it is a hypothetical situation. A cell is never at rest, it is always dynamic. It will be doing some of the other function, either its metabolic function, it will be producing some proteins, if it is a neuron, producing some neurotransmitter, etc. So every time the cell is active. So I'm talking about a hypothetical situation where the cell is at rest. And in that hypothetical situation, if we are moving into the nucleus, and if we are looking for the structure of the genetic material inside that particular nucleus, you can see that the DNA, or the genetic material, will be in the form which is called as the solenoidal structure. It will be so if it is a human cell, all the 46 chromosome or 23 pairs will be present in the form of solenoidal structures. It will be in the form of solenoidal threads, and that starts to form a chromosome only during the particular different stages of division in either mitosis or meiosis. Otherwise, the DNA is seen in the form of solenoid, in the form of a solenoidal structures. So now in this particular session, we'll start with discussing the significance of this particular structure, or why this DNA is seen in this particular structure, which is called as a solenoid. So now we'll see how, or what is the significance of this particular structure called as a solenoid, or why the DNA is maintained in that particular structure called as solenoid inside the cell, rather than the normal double helical loose structure. So one is the reason that I said during the discussion of the structural organization, why it is seen in this particular form. That means we have to store that much amount or lengthier amount of DNA in a very small nucleus. So that is one reason why the DNA is seen in the solenoidal structure. The second reason is the most significant reason for compacting the DNA in this particular structure called as the solenoid. And we'll move on to that particular important reason for this, or the significant reason for the solenoidal structure. So the reason is nothing but we know that the main function of the genetic material or the DNA is to produce proteins via the particular pathway which is called as the central dogma. That means central dogma is nothing but, you might have heard, if you have not heard, uh, uh, we are going to discuss in detail in the next semester, in the sixth semester, about the central dogma. Central dogma is nothing but a DNA. It transcribes a particular strand or stretch of DNA into an RNA, which is usually called as an messenger RNA or an mRNA, and that will be translated into a protein. And this particular two-step process, transcription followed by translation, is called as central dogma of molecular biology. So in this particular central dogma, that means a DNA, the main function of the DNA, that means we have studied in lower classes that the nucleus or the DNA or the genetic material inside the cell is the controlling unit, or the one which controls a cell. The nucleus is the controlling point of the cell. So how the nucleus controls a cell is by producing mRNAs. Nucleus or the DNA can do nothing other than that. It produces many different kinds of proteins through the mediator molecules called as mRNA. That means DNA produces mRNAs, that mRNAs will be produced into the, or it will be translated into the proteins in the cytoplasm. So this particular step of central dogma happens inside the nucleus, and this mRNA to protein that happens inside in the cytoplasm. So this particular link between, or this is how your nucleic acid is controlling the cell. It produces different kinds of protein. Whatever the protein is present inside the cell is produced by the DNA through this particular cycle. And it is that proteins which is controlling the cell. Controlling the cell means these proteins might be enzyme for reactions, various enzymes for the reactions. These proteins might be the structural proteins which is forming the plasma membrane, which is forming some cell organelles, etc. So that is how the nucleus is controlling the cell. So we were talking about the solenoidal structure and the significance of the solenoidal structure. So we said that the major or the one and only function of DNA is to produce proteins by controlling the cell. And every cell in our body, that means if you take a cell in your fingertip, if you take a cell from your hair follicle, etc., every cell in our body is having all 46 chromosome or 23 pairs. That means every genes that is required for our body is present in every cell. But we know that hair, which is a protein which is called as keratin, that particular structures or the hair is produced only at certain locations of our body. For example, in our hair for males, in the beard, mustache, etc., in your armpits, etc. Only in such location, the particular or the cells produce that particular protein called as keratin, not anywhere else. And for to take another example, only the pancreas produces the protein called as insulin. The insulin gene is present in the hair follicle cell. So now we come to the question, why insulin is produced only by pancreatic cells, and why not the hair cells or the hair follicle cells produce insulin? Because insulin gene is present in the hair follicle cells, keratin gene is present in the pancreas, but you don't see hairs in the pancreas, you don't see your follicle cells producing insulin. And this particular different, that means one particular cell produces particular protein, the other cell is producing particular protein, and this particular difference is called as, the term is called as differential gene expression. So we'll come to that, what is this differential gene expression, etc. So this particular difference, that means A particular cell produces a particular type of protein, but the other may not produce that particular protein, and this difference is termed as differential gene expression. And now we are going to see what is the relation between differential gene expression and your solenoidal structure. So it is a solenoidal structure which determines differential gene expression. So now we'll see what is this particular thing which is differential gene expression, etc. So gene expression is nothing but we'll talk about gene later. So for this discussion, we'll put the term called as gene. So gene is nothing but a fragment of your DNA, a fragment of your double helical DNA, which is capable of producing a protein. So a fragment of a DNA which is capable of producing a protein means it is capable of producing an mRNA. So from this term itself, it is clear that the entire DNA is not capable of producing protein. So there are some locations in your DNA strand which can produce a particular mRNA, and that mRNA will go on to producing a protein. So those regions of your DNA which is producing an mRNA is called as a gene. Is that clear? So in every cell there are genes. That means we have 23 chromosome. In all the 23 chromosome there are genes which produces different kinds of enzymes, different kinds of proteins, etc., different kinds of protein hormones, etc., which are required for our body. And the thing is, what we said earlier, which is called as a differential gene expression and the solenoidal structure. That means why your hair cells do not produce insulin. That is because of this particular structure called as solenoid. That means usually what happens in an protein production is that you have this how a protein, or now we'll see how a protein is produced, etc., in a very brief manner. So you have a double helical DNA like this. Okay, on this double helical DNA, some proteins which are called as, or enzymes which are called as polymerase enzymes, which are called as, usually called as RNA polymerases, they bind to it, and this RNA polymerase, after binding to this particular double helical DNA, it is capable of producing mRNA. Okay, so this produces what is called as the mRNA. That means whenever this particular our RNA polymerase can bind only to double helical DNA. It can bind only to a structure which is called as a double helical DNA producing an mRNA, and that mRNA will move on producing that particular protein. So what happens inside the nucleus? So now we'll talk about this differential gene expression. So from this, it should be clear that a protein or an RNA polymerase, to produce an mRNA, it should bind to a DNA which is in the double helical structure. So we said earlier that inside the nucleus, our DNA is present in the solenoidal structure. That means it is tightly compacted. In that tightly compacted form, so I draw this as the solenoidal structure, which is compacted, etc. So this is the inner coils which are present with the hexagonal structure, which is more and more compacted. This is a solenoidal structure. So in this solenoidal structure, I said about a protein which is called as the RNA polymerase. So if your DNA is present in the solenoidal structure, your RNA polymerase cannot bind, because RNA polymerase will bind only to free double helical DNA. Otherwise, in molecular biology terms, it is called as a naked DNA. Naked in the sense it is not bound by proteins which are called as histones. If a particular DNA structure is bound by histones, it will attain the solenoidal structure. So whenever these histones move out from the DNA structure. Such DNA or such double helical DNA is usually referred to as a naked DNA. That means without the protein. So only to such naked DNA RNA polymerase can bind and produce that mRNA, moving on to producing that protein. Is this clear? So if this is clear, the next important thing is our first question: why not hair follicle cells produce insulin, even though it is having an insulin gene? So this is the reason. So I take this particular solenoidal structure itself. So if this is a DNA or a compacted solenoidal structure, and here I'm having the gene take for example, this is the region where the DNA for insulin is compacted into the histone. So this is for insulin, and in the same structure at another different location you have the particular uh, gene or the DNA segment for keratin. Now it is in the packed state, which is called as a solenoidal state. Your RNA polymerase cannot bind. And consider this particular solenoid in your hair follicle cell. In the hair follicle cell, what happens is, inside the hair follicle cells, there are some other factors or there are some other enzymes inside the cell or inside the nucleus which will modify the solenoidal structure. So modify the solenoidal structure in the sense that it will modify the particular gene which is going to be expressed in that particular cell. That means if this is a chromosome inside the keratin or inside the hair follicle cell, what happens is that there are some enzymes which will come and modify this particular site of the chromosome. Modify in the sense that they will inhibit the histone molecule. They will try to remove the histone molecule. So whenever the histone molecule is removed, so whenever the histone molecules are removed from here, what happens is that whenever the histone is removed, the double helical DNA, which is also referred to as the naked DNA, will loop out. It will not break from it, because a DNA means it is a continuous strand from one end to the other. You cannot break the DNA. If a DNA is breaked, that results in mutation. So DNA is never broken inside the cell. So this is the process which is called as looping out of a DNA. Just imagine you have tightly coiled or you have a roll of thread, and try to take a particular thread from not from the ends, from the middle. That means you are taking or you are pulling out a strand. You pull out a small portion of that thread outside. So in such a particular structure or such a thread, that means it is now free from that compacted roll. A small portion of that thread is free from that compacted roll, and that particular structure can be taken analogous to this looping out of the DNA. And now when this particular DNA is looped out, what happens is, inside your nucleus there are proteins which are looking for naked DNAs to come out. So whenever a naked DNA comes out, it will be the site of attachment for the so-called RNA polymerases. If this is the keratin gene, it produces the protein for the mRNA for the protein called as keratin. It moves to the cytoplasm, produces keratin protein, and that results in the formation of hair only in the cells of hair follicles. The same happens with the pancreatic cell. The pancreatic cells has both keratin gene and insulin gene. There the opposite happens. That means the factors inside the nucleus will modify this particular site of the chromosome. As a result, this particular keratin site, so if I erase this, so in such a case, what happens is, if it is a pancreatic cell, this particular DNA region will loop out, RNA polymerase will bind, produce mRNA, and that mRNA will be the mRNA for insulin, which will happen only in the pancreatic cell. So this is why, even though every cell in our body is having every gene is present, because every cell in our body, even the cell which is present in your fingertips or in your toe tips, are having every gene, because we have the 46 chromosome in every cell of your body. In every genes are present, and in that condition, or even in such situation, we are having specific genes which are being expressed at specific location. uh, Gene expression or protein expression means nothing but, gene expression and protein expression is the same. Gene expression means production of a particular protein, is also referred to by the term called as gene expression. So this is what is called as differential gene expression, which is mediated by the solenoidal structure. So solenoidal structure is the one which is inhibiting every gene from expression. It will not allow the RNA polymerase to bind. If only RNA polymerase binds, mRNA can be produced. If at all mRNA is produced, that will form a protein. So here differential gene expression occurs because of certain factors which are present in specific cell. That means the factors which are present in the pancreatic cell will modify the histones in the insulin gene. The factors which are present in the hair follicles will be modifying the gene region for keratin. That is why even though these two cells, in our example, the keratin or the hair cells or the follicular cells and the pancreatic cells, even though they are having both the genes, pancreatic cells produces insulin and the keratin or the follicular cells produces, or the hair follicular cells produces keratin. So this is the significance of this particular structure, which is called as keratin, and that is why we are having nails only at our fingertips and toe tips and not elsewhere. In normal cases, there are abnormalities. In normal cases, our finger, the nails which are present only in our fingertips and our our toes. So these are the examples, that is specific, the formation of specific proteins by specific cells, even though they are having genes for every the genes for every chromosome, or genes for every not every chromosome, genes for every protein inside our body. So every cell in our body is having the genes. It is this differential gene expression that is mediated by the solenoidal structure. We are having different structures at different parts of our body, or different proteins are being produced at different regions of our body. So this is the significance of the solenoidal structure. This was the main significance of the solenoidal structure. And the first one, as we said earlier, the significance of the solenoidal structure, the first one was to compact the DNA to fit into the nucleus, and the second one is this is a structure which allow differential gene expression. That means specific proteins are produced only by specific cells, because it is inhibited by this particular solenoidal structure. So with this, we come to the end of this solenoidal structure, and we are moving on further. So this was our discussion with the solenoidal structure, etc. And now we are going to see the details of the other terminologies, like the most confusing terminologies, like chromatids, then chromosomes, sister chromatids, non-sister chromatids, homologous chromosomes, etc. So we'll discuss this particular confusing terminologies, because these are the most confusing terminologies when you come to genetics, when you come to molecular biology, etc. So we'll try to solve this confusion between these terminologies. For that, we'll be starting with this particular thing, which is called a cell cycle. So we'll see that in the following video. Thank you for listening.